Okay, so thanks, Bella. Thank you all for the opportunity to present our ongoing work focused on the early stages of X-linked juvenile retinoschisis. X-linked retinoschisis is a prevalent early onset disorder for males, less so for females, of course, due to it being on the X chromosome. But even though X-linked retinoschisis can be diagnosed at a relatively early age, the window to study the very early phenotype is a moving and vanishing target in humans. So to address this, we've been using mouse models. But first, for those who are unfamiliar with this disease, this slide captures three of the main features. When you look at the retina on the fundus, whoops, I guess I can't use my pointer. When you look at the retina through the pupil, a trained eye can see the foveal cysts that create an unusual spoke wheel pattern. Those are pointed out by the large arrowhead. Second, as shown on the right, the ERG is abnormal, with a greater reduction for the later onset B wave than the initial A wave. This reflects a defect in transmission from photoreceptors to the bipolar cells, which actually generate the B wave. Now, with the advent of optical coherence tomography, the large intraretinal cystic skitic cavities are clearly present, as shown on the lower left. About 25 years ago, mutations in a gene named retinoschisin were identified in patients with this disease. Note that the reason that the gene and protein were named after the disease is because both did not fall into a readily available classification system. Now, this is a complicated diagram, but the main points are that formation of mature retinoschisin protein requires several steps, from initial translation and folding as shown on the top. Can you see my mouse?
Yes.
Oh, well, that's exciting. I'll use it. From initial translation and folding to forming a single RS unit here. This is a monomer, and these monomers converge to form a octamer with 8 subunits combined into a single unit. And then these are secreted, and extracellularly, 2 of these form a double octamer. That's the mature form of the protein. Now, shown on the bottom, disease-causing mutations occur throughout the RS1 gene from the first exon to the second exon and through to the entire gene. And these impact the initial folding process. They, in fact, impact the formation of the octamer and various other steps along the way. Now, a few years ago, the good folks at Regeneron Pharmaceuticals called me about working with them on 3 new mouse models for X-linked retinoschisis that they were developing. The mutants were chosen to impact different aspects of retinoschisin formation. So the knockout was chosen to cause complete deletion of the protein. The C59S point mutation was chosen to prevent formation of the octamer and to mirror a human disease mutation. And the R141C was chosen to impact protein formation, also a human disease causing mutation. That's peeped up there. So we were not the first group to work with RS1 mutants, but most work that's been published to date has focused mainly on the adult phenotype and how that was affected by gene replacement therapy. We focused on the earliest stages of retinal development. Now, the top panels shown here show the time course over which the RS1 gene expression is developed. So it's first seen really at postnatal day 5, and then even at later ages, all the RS1 expression is limited to the photoreceptors. The lower panel shows where the protein goes. Although expression is restricted to photoreceptors, RS1 protein is distributed throughout the retina. And that's consistent with this protein being secreted by photoreceptors to go do wonderful things throughout the retina. Now, these images show where the mutant proteins end up in the adult retina. In the control, retinoschisin is seen throughout the retina. In the knockout, we do not see any retinoschisin, which simply demonstrates that the knockout works. Note in both the C59S, as shown in the lower left, and the R141C, there's a decreased overall level of the protein, and it has a very abnormal distribution limited to the inner segments in the C59S and spotty expression throughout the retina in the R41C. So here's our data. These are OCT images taken from very young knockout mice and wild type comparison. what we were impressed to see was that at postnatal day 15, which is a pretty young mouse, there's remarkable levels of schisis in that animal compared, which is not present in the wild type. And what we also noted was that over time, as the mice aged, the level of schisis became less severe, although it certainly did not disappear. We also noted very marked ERG abnormalities. Now, this is another busy slide, but if you just focus on these data P15, the red lines, which are obtained from an RS1 knockout, are markedly reduced in comparison to the wild type controls. Now, our ongoing work is moving to even earlier ages. What we've seen is that at P13, we can see clear evidence of schisis in all of the mutants, the knockout, the C59S, and the R141C point mutants. So that's a P13. It's not as bad as the P15, but it's clearly there. At P11, things look pretty good. So we don't see any schisis. So it seems like we've identified the window at which schisis presents. So just to summarize, our mouse models develop a very early disease phenotype that is not dependent on the specific genetic mutation. It's similarly, we believe it's likely that the human phenotype also starts very early, but it's very hard to get human patients at these young ages for these types of studies. What are we doing now? Our current work is focused on the possibility that normal photoreceptor development is never achieved. While we do see fairly normal retinal structure at postnatal day 11, we're now using calcium imaging to examine the function of single retinal cells and the ERG to examine overall retinal function. In parallel, we are measuring vision in X-linked retinoschisis patients. Our approach is based on prior work showing with retinopathy of prematurity, which shows that the developmental refinements that pertain to our abilities to see spatial stimuli, to see visual stimuli changing in time, are never fully developed. And whether this is the case for patients with X-linked retinoschisis is one of the questions that we are trying to address. Finally, we'd like to understand the role of retinoschisin in the adult retina. The early onset degeneration that we see in our mice precludes really understanding what the protein might be doing in the adult retina. So to address this, we're working with the Hackstrom lab to develop a mouse model in which retinoschisin is normal through development, but only at adult ages is it taken out genetically. And I just want to acknowledge our collaborators. This work reflects contributions from several labs. We have an NIH award that supports our work here in Cleveland, and these are the folks who worked on this project in Cleveland. The grant also supports work in Chicago. This is Jason McEnany's lab, who is working on the human psychophysical studies. And also there's work going on at Cornell downstate in the lab of Botir Sagdaleev. And I just want to point out that this arrangement here runs almost through my full career. My first paper as a postdoc when I was at UIC was with Jerry Fishman, and it was about patients with retinoschisis. So I think I've run my entire career circle to its end. That's sort of terrifying. Anyway, and before I close, I just want to acknowledge our ongoing and very enjoyable collaboration with Carl Romano and his group at Regeneron Pharmaceuticals. Thank you.